material. We have several days that I've set aside at lab time to do reviews and so you don't forget the brain as if you could. But we will be adding some new material such as the eye and the ear components, but we do have labs that we can go back and review. So this unit right now, the 2.5 weeks that we have on this unit is the most important in my mind of everything that we have for this entire semester as far as what you're going to use in other classes. We're going into nursing, next semester, Bio 431, this neurophysiology and the channels that we talk about. We're going to keep bringing back in the autonomic nervous system. We're going to keep bringing back in second semester. The major reason why I make it an open book on the exam doesn't make it easier, but it means that I can ask you questions on concepts that I don't expect you to have memorized. All right, there's a lot of different neurotransmitters, there's a lot of different receptors for those neurotransmitters, pathways by which they're taken up and broken down that the first time through I don't expect you to be able to pull up from your brain and be able to answer off the top of your head. Because of that, though, because it's a take-home, expect that you're not going to find every answer in your lecture notes or in the textbook. Okay? There are some material where the concepts are there, but the specifics you're going to have to look up. So, in other words, questions, I might ask about the toxin from a pufferfish. I might ask about for someone else's test, because you don't all have the same exam. It might be a toxin from a black widow spider or a black mamba snake or something like that. So same general concepts, but specifics are different. For that, because various online sources are different in their accuracy, in some of these things new information is coming out. It would be helpful for you if you dispute if I mark an answer right or wrong, to keep a log of where you find the answers. Okay? And turn that in with your exam if you wish. The exams will be available next week online, and I will pre-assign you. There's 5 or 6 different ones, and I will cluster you between the morning and the afternoon groups. So you'll find your name online and print off that particular exam. Okay? So the exams will be on D2L for you to print off. They are due, our lab exam is on the 19th, so I think that's a Tuesday. The lecture exams will probably do the end of that week. You'll have about 3 weeks, starting next week when I get them to you approximately. Also, however, we still have online quizzes over this material. All right? So you do have to learn it to some degree, but I'm going to change the time available from 15, you have 20 minutes currently, I'm going to change it to 30 minutes. Okay? So instead of 15 minutes, 20 minutes to do 15 questions, you'll have 30 minutes. All right, but you still need to study it before you take the quiz. It's not an hour that you can look up every single answer kind of thing. All right, so we're starting off with neurophysiology. Tuesday is scheduled to be autonomic nervous system, but we're actually going to finish neurophysiology because I've added some additional autonomic components to the neurophysiology and the neurotransmitters that we're going to kind of flow into the autonomic nervous system lecture component. And tomorrow is not a lab, it's all lecture. So you will get out a little bit earlier, but I'm going to lecture longer. Does that make sense? I'm not going to lecture for the entire 3 hours, but I'll lecture more than I usually do, and with breaks it'll be a little bit earlier than you normally get out. It won't be a lab time. I'm not going to lecture the entire time, however. Let's just do a little bit of review of what we've covered so far on the neuron and the physiology of the neuron. Because I actually talked about this a little bit when we talked about the structure of a typical neuron. Because today we're going to look at different types of channels that are available. So I just want to do a short review, not where your lecture notes start, but a short review on the channels we've already talked about. So what do we mean by an ion channel or a protein channel? Remember way back when we were talking about membranes? Okay, so why do we need a channel for an ion in our neural cell membrane? All right, the major type of chemical bond that we find in our plasma membrane is from the lipid. That's what most of the region of the membrane is, and that's a nonpolar covalent bond for the lipids. Our ions are charged. They were involved in ionic bonds. And so they're not going to move through the lipid material, through the fatty acids. So the first couple of pages of your lecture notes is something I found, an article I found a year ago, taken from Ion Channels for Beginners. It provides a really good analogy of how ions need to move through ion channels. And it kind of explains it in a manner as if you fell into a pit, and to get out of that pit, you have to climb up rungs of ladders that are built into the wall. So the pit is the ion channel. You're the ion. And the ladder rungs are the positive and negative charges that allow an ion to move through. And it talks about how as you move up, the channel or the pit gets smaller, and you have to take off a jacket or you won't fit through the opening. And so ions are forced to shed. They're attracted because they have a charge. They attract water molecules. Water molecules are polar. And so as they move through this channel, either into the cell or out, they have to shed these water molecules or they don't fit. And the protein components, the positive and negative charges of the protein forming the channel, change, and they interact with the ion the way the water molecules would have. And that's kind of what helps keep the ion moving through the channel. And then, let's say another ion comes behind it. So if you're climbing up the ladder and there's someone behind you and they're saying, hurry up, all right, or they're coming down and they're stepping on your hands, you're going to move down the ladder faster. So as another ion, let's say I have a potassium ion moving through a channel, another positive potassium ion coming along behind me is going to have that electrode, it's going to push me away, because two positives don't attract each other, they push away. And so another ion going through the channel helps to move the first ion through. Anyway, it's just I don't test specifically on that, but it does provide, I think, a good, it's hard to think of abstract components when we're thinking of tiny, tiny little ions. And so the analogy that's made here, I think, is helpful in understanding how these ions are moving through. So when we first started talking about channels, when we talked about the structure of a neuron, I just basically told you ligand-gated channels were found in the cell body and dendrites, and multi-gated channels were found in the rest of the neuron. So let's kind of go back to that point, review that concept, and look at other types of channels. So we have the synaptic bouton of a presynaptic neuron. And it's going to form a synapse with the dendrite of a postsynaptic neuron. That's what we call the communication between two neurons, is a synapse. So this synaptic bulb has been excited to release neurotransmitters because of a current coming down from its previous component, and these neurotransmitters, the vesicle fuses, and in the dendrite of our postsynaptic neuron, we have channels for ions that are closed normally, and when this neurotransmitter binds to it, that channel opens, changes shape, and opens to allow ions to move either into the cell or out of the cell. On tomorrow and on, well, primarily tomorrow, we'll talk about what happens depending on the type of ion that moves in. If these are sodium ions that move in, all right, then the inside of the cell is going to become more positive. And depending on the number of synaptic boutons or the number of neurons that synapse with this cell body, the more open sodium channels we create, the more positive ions are going to be inside the cell. So if we look at the rest of the cell body, we have the axon, and in our axon hillock, and all of these ions are going to flow towards and away from their original location. They're diffusing. Simple chemical, simple diffusion. High concentration to low concentration. Now, because these ions can be leaving the cell and moving out to the other dendrites, there's nothing that says they have to go straight to the axon. So if they're coming in from this particular dendrite, they can go out this one, over here, over here. They don't all have to go towards the axon. So the number of ions that get to the axon are going to be fewer than the number of ions that came in right here. But let's say that we have other neurons forming synapses with these other dendrites, and all of them are opening channels, allowing ions to come in. And we're going to get a pretty strong flow of charged particles by the time it reaches our axon. So what do we call a flow of charged particles? Do you remember what form of energy that is? Electrical. Okay, it forms a current, electrical energy, that we measure in millivolts. So here's an example of a ligand-gated channel, or chemically gated channel. And as you'll note, because we're going to be talking about potentials tomorrow, I can have a weak current if I only open one channel and allow 25 ions in, or I can have a strong current if I open 100 channels and allow 2,000 ions in. So this channel can create a variety of strengths of currents. Okay. If that strength of current is strong enough, when it reaches the axon, then I can open a different kind of channel known as a voltage-gated channel. That one does not interact with chemicals, but it interacts with the charged particles. And as it binds to those charged particles, that changes the shape of the protein. Usually opens it. It can close it, but usually opens it. So if sufficient particles come by, then that channel will open and ions will either leave or come into the cell depending on what kind of a channel it is. So these are the two types of channels that we were just barely touched on when we looked at the structure of a neuron. I'm going to go back over those today and look at two others. But because of the voltage-gated channels, we start forming a type of current known as an action potential. So I'm just going to give you the big picture here. This creates an action potential, whereas our ligand-gated channels created a graded potential, meaning it could be weak or it could be strong. Action potentials are always the same strength every time it's made. As this action potential travels to the end of the axon, it reaches the axon terminals and the synaptic boutons of this axon, causing it to release neurotransmitters, and we can continue this whole process. Okay? So what we started at the dendrites and cell body, we can continue with another synapse with another neuron. So let's look at these four basic types of channels. They're listed in your lecture notes. The first one we're going to talk about is a channel that's open all the time. Can I erase the board? That's where we're at.
No.
Okay. So weak channels can be for a variety of ions. The most common ions that we're going to be talking about are primarily potassium and sodium. We'll also talk about calcium, a little bit about chloride, and a little bit about magnesium. Okay, yes?
For those voltage-gated channels, in that instance right there, are those also lead and sodium ions?
Some voltage-gated channels are sodium, some are potassium,
So
some are calcium.
if the synaptic bulb releases sodium ions and they diffuse and go into the channel, will it also be introducing sodium ions, or is it okay?
The sodium, the synaptic bulb only releases the chemical. It only releases the neurotransmitter.
Okay.
It will bind, the neurotransmitter is specific, so if it's acetylcholine, it will only bind to acetylcholine channels. Now, depending on the neuron, those channels might be, they're often, most of the time, they are for sodium. They can be for potassium, but usually, I'll talk in just a second, usually in an indirect mechanism are they for potassium. These ion channels, when we talk about the leak channels, they are specific. So potassium leak channel may allow a sodium ion in, but at a ratio of about maybe 12,000 to 1. So one sodium ion may pass through for every 12,000 potassium ions that pass through, and it's going in the opposite direction. So there are other channels that will allow both sodium and potassium to go through. So we're going to be talking about that a little bit more specifically when we talk about neurotransmitters and exactly what they do. Does that answer your question? I'm not quite sure what you were asking.
I'm pretty, well, I was, let's wait and see.
Okay. I'm going to be talking about voltage-gated channels again, so that might help to answer that question. I just wanted to kind of tie us back to where we'd already talked about them and where in the neuron we're interested in their performing. All right, so a nerve cell, or any cell, but we're looking at nerve cells right now, possesses leak channels. Now, they used to be called leaky channels. I'm not sure why somebody decided to just drop the line and call them leak channels. So if you see me saying leaky, it means the same thing. Leak channels are leaky. And they are typically preferential for one particular ion. And they are open all the time. They never close. Okay? Most of our channels are gated, but we have sufficient leak channels that they can affect the overall charge inside the cell as compared to outside the cell. So a leak channel for sodium, sodium is in higher concentration inside the cell or outside the cell? Well, there are other factors that are going to affect this. Depends on the number of channels, and we also have our pumps that are going to come into play. But in the cell, in the body, because of these other factors, we have a greater number, more than 90% of sodium ions are found outside of the cell. Right? And so, more than 80% of potassium ions are found inside a cell. So if we're talking about sodium, and we have a few leak channels, then the direction the sodium is going to leak is going to be into the cell. So we have more positive ions outside the cell for sodium, a few sodium ions inside, and so the direction of diffusion is into the cell. We also have potassium leak channels. Again, leak channels mean they're open all the time. And we have more potassium leak channels than we have sodium leak channels. So because of that, we can make the statement that the cell is more permeable to potassium. It's not that it moves across through the lipid molecules any easier, but it can cross the cell membrane easier because it has more transport sites, not transport, but more availability to cross. We have more sodium ions inside the cell. I'm sorry, more potassium ions inside the cell. And so with our leak channels, potassium is going to leak out of the cell. So if we look at a leak channel, the blue is the sodium leak channel, and there's more potassium leak channels than there are blue, that's the yellow one. And on the far right is the voltage meter. So it's indicating that the inside of the cell is slightly more negative than the outside. And we'll look at why this happens in a few minutes. So if we click on a sodium leak channel, there's no pump moving it in, there's no gates that have to open, it's just going to move in by diffusion. What's going to happen to the number of positive charges inside the cell? Overall, if we just look at a sodium leak channel, inside the cell is going to become more positive. Okay? Now let's open our potassium leak channels. And you'll see the needle drifting over towards the negative, over towards the left, as the inside of the cell becomes less positive, or more negative if you wish to prefer it in that direction. So this is going to affect the difference in charge between the inside of the cell and the outside of the cell. What we're leading up to is we have to have a difference in charge to create a resting membrane potential. Otherwise, our action potentials can't be created. So that's a leak channel. Now let's look at a chemically gated or ligand-gated channel, which we've already talked about. And these can be either direct in function or indirect. And it's a matter of timing and control. May I erase the board? Now next week we're going to start talking about the autonomic nervous system. And one of the parasympathetic, we've mentioned the oculomotor nerves and the facial nerves and glossopharyngeal and vagus are parasympathetic. So one of the concepts I'm going to introduce to you, let's look at vagus. Vagus comes out of the medulla. And it has a motor neuron in its nucleus in the medulla. Let's say it's going to the heart. All right? So it sends a long, I'm going to draw this midline, sends a long neuron to the heart. And inside the heart wall is a second neuron that actually goes to the cardiac muscle tissue in the atria. So we actually have two neurons rather than one single lower motor neuron, but we'll get to that next week. I'm going to take this area right here and enlarge it. Our neurotransmitter happens to be acetylcholine, right? Between from this neuron to this neuron. From this neuron to the cardiac muscle, it also is acetylcholine. However, those are both ligands, those are the same neurotransmitter, but they work in two different mechanisms. This one is fast, always excitatory, and direct. This one is slow. In the heart it happens to be inhibitory. A similar synapse in the stomach would be excitatory. So this one can be variable. And here's why. Okay? So in the direct pathway for ligand-gated channels, we have our plasma membrane. This is outside, this is inside. And we have a receptor, a molecule, a protein molecule, that recognizes and binds the neurotransmitter. All right? When it is not binding to that neurotransmitter, our channel is closed. Here it's shown as a tube. When it does bind the neurotransmitter, it changes in shape. And the receptor, the protein that binds the neurotransmitter, is actually the ion channel. And here it is open. So what I mean by direct is the molecule that binds to the neurotransmitter is the ion channel itself. And that's the way we've been describing it so far. Okay, this acts very rapidly. And that's what's illustrated here. Neurotransmitter binds, the stereometric interaction causes the protein itself to change its shape, and the ions go into or out of the cell. All right, that's what takes place, as we'll see in a few days. That's what takes place here. However, at the end of the second cell, we have an indirect pathway. This can take 2 or more steps. In this case, the molecule that binds the neurotransmitter is not a channel at all. It is just a receptor. Associated with it is a series of molecules known as a G protein. And again, we have a whole other series of talking about this. When acetylcholine binds to it, it changes shape, it activates the G protein, it separates part of that G protein, moves down the membrane, and now it can either directly open a channel or it can activate enzymes inside the cell that those open the channel, or a variety of events can occur. So this can either open the channel, it can activate enzymes that open the channel, or it can start a cascade. But downstream from this is our actual channel. So the molecule that binds to the neurotransmitter is not the actual channel. And that's what we mean by an indirect pathway. This concept that I just touched on here that we will come back and spend more time talking about is going to help you immensely the first week of Bio 431, because that's what we do, is talk about that again. All right, we're going to have that first lecture of Bio 431 in this unit. So it should be just a repeat and a review of it. But the direct and the indirect pathway in both cases requires a chemical, and that's what we mean by ligand-gated. Okay. The advantage of this is that it's fast. But anything that mimics acetylcholine is going to result in these channels opening. Disadvantage is it's slow, but we have multiple areas of control. We also have amplification. One neurotransmitter binding here can result in 20 of these or more channels opening. So we can amplify the result. Okay? So that's a ligand-gated channel. Then we have the voltage-gated channel. May I erase the board here? No, not yet. Ligand-gated channels can create a variety of strengths of current. Okay? We can open multiple ligand-gated channels and create an increase in the amount of ions. If not very many ligand-gated channels open, then not very many ions are created. We can make the inside of the cell less negative, more negative, depending on what kind of channel we open. So ligand-gated channels provide a great variety. When we talked about Parkinson's disease, dopamine was an excitatory neurotransmitter and acetylcholine was an inhibitory. In other situations, acetylcholine is excitatory. So we'll look at how that can happen, and it happens because of those indirect pathways. Okay, can I erase it now? All right. Primary function of voltage-gated channels is to send the signal very rapidly down the axon or long process of the neuron. Well, ligand-gated channels are at the synapses. That's how one nerve cell communicates with another nerve cell. But then when we want to send that message, down the axon, or the central or peripheral process of a sensory neuron, the voltage-gated channel functions much more rapidly because its strength of its current is constant. It's not going to dissipate. And remember the analogy I gave to dominoes? Once we push one domino over, that's our voltage-gated channel open. That force of the domino falling is sufficient to knock the next domino over. And so each voltage-gated sequence opening creates a current strong enough to open the next voltage-gated channel. So it's self-perpetuating in that sense. It doesn't create its own energy, but it provides the source of energy required for the next channel. And so this is what we use to send signals from sensory neurons all the way up to the spinal cord, or spinal cord all the way up to the brain, and vice versa for motor channels. All right, so if we look at this image here, these paddles, the light and the dark brown, are indicating the two positions of open and closed. All right? So in this case, with the dark brown that you see here, the channel is open because a current is going by. When the channel is closed, the paddles are in this position. So I'm going to diagram that on the board, but before I do so, as the current is going by, it's interacting. Remember, positive ions are attracted to negative ions? And so those ions interact with the opposite charge on the protein, and there's only a maximum number of opposite charges that it can interact with or affect. So once we reach that, sending even more charges down by the protein isn't going to change it. I alluded to this when I talked about opening, walking up to a department store or a grocery store. And they don't want, city rats running by, or country rats for that matter, to be able to open the door. So you have to have a minimum weight or height if you're breaking an electric eye. If you're standing on a mat, they don't want a 10-pound whatever opening it, so 30 pounds or whatever to open the door. If you weigh 500 pounds, the door's not going to open any faster, okay, once that trigger has been activated. So if we have a voltage-gated channel in its closed form, so use this illustration here. This would be closed. And then we send a charge. All right, we send ions by that have come in via our voltage-gated channel. And as these ions flow by, remember the movement of charged particles is how we define an electrical current. As they interact with this protein, that causes it to change shape, and now it looks like this. Again, this is just an analogy, but it gives you a visual. So these move, and that creates a channel depending on which way the ions are going to flow. So it's not that both of voltage-gated and ligand-gated require the protein to change shape. That's what opens the channel. All right? It's what causes that protein to take shape that is different. Either the chemical recognizing a specific region on the molecule as a receptor, or the current going by. Okay? Now the third one on there isn't as common, and that's the mechanically gated channel. I don't have a diagram that illustrates that. But for an example of a location in your body that uses mechanically gated channels is your ear. Remember I talked about the fluid in the ear? If you start to move, that fluid flows, or if I tilt my head to one side, that fluid is going to move. and it's going to bend very long microvilli as it flows by them. All you have to do is look in an aquarium and see the current flowing over anemones or something. It's going to cause it, well, anemones do their own movement. A plant, it's going to cause it to move. Okay? So with the cell membrane structure, when that microvilli bends over, it stretches the membrane. If you were to take one of those long balloons that you make animals out of, and you bend it, that balloon, that latex stretches. When that occurs, these mechanically gated channels open. All right? And current is created as ions flow in or out. Now, in my eye, I have rods and cones. Their channels are activated by the energy of the photon passing through my eyeball and going to the rod and the cone. I can change that to mechanically gated. I close my eyes and put pressure on my eye. I can create a play of lights on the inside of my eye just by the pressure I'm putting as I change the voltage by mechanically opening the membranes of those cones. All right, and that's sending a signal back to my occipital lobe, which is projecting it back where the pressure is, which is my iris. But I'm activating that first electrical current through mechanical deformation of the membrane. So there's a few areas, not a lot, in the body where that is effective. So now let's look at potentials. Okay? For some students, the word potential can be a little difficult to understand. So if that's the case, change it to current. By the word potential, we mean there is the possibility of ions flowing from one side to the other side of a membrane. And so we will use several terms. We won't finish it all today. We'll use several terms: resting membrane potential. Without a resting membrane potential, we can't create the other membrane potentials. Graded potential I've already used. That is a change in the resting membrane potential that can be small or large, so we can grade it. Okay, like a piece of meat. Yeah?
For the voltage gated one,
Uh-huh.
do those, the paddles there, do they go from negative to positive, or were they just...
Well, adding the interaction from the negative charge to the positive charge is what changes. So the positive ion would bind to, that's one theory, would bind to the negative region of the paddle, causing it to change shape as its pH changes. Changing the pH around a protein can cause that protein to change its shape. So some proteins have a better shape. That's how we can have enzymes denature with different pHs. Okay. Some enzymes in our body won't work at a pH too high or too low because it won't recognize its substrate. All right, so let's look at, first of all, how do we establish the resting membrane potential in our nerve membrane. So you have this diagram, let's see, right here on your lecture notes. So the resting membrane potential is shown here in the cell body of the neuron, but it actually is found across the entire neuron. We change that resting membrane potential if we create a graded potential. We change that resting membrane potential if we create an action potential. All right, it's our baseline, if you will. So this is the cell membrane of the neuron, either here or at the axon. These are showing some of the different channels that are present and ions that are present inside the cell. Now, to measure the difference in charge, we have to measure the concentration of ions inside the cell and outside the cell. This uses a voltmeter. And the way this was started, now they have, different types of machines. But the basic machine, I don't know if any of you have ever seen a glass pipette. We tend to use plastic ones because they're disposable. But glass pipettes, any pipette has this shape where we start out here and then we go thin. All right? And thank you. But the plastic pipette, it comes with its own bulb, just like you were using when we did the cut out the little circles in the auger for the diffusion. In the glass pipette, you have to add a rubber bulb. But if this is heated over an open flame, like a Bunsen burner, you can take that glass pipette, like you were playing as a glass blower, and just stretch it. And the more you stretch it, I done this with them, it becomes as tiny as a hair and it's still hollow inside. You just keep pulling and pulling and pulling and pulling. Obviously it breaks easily, so these are best made with an instrument, not two human hands, but a machine that pulls them apart. And you can create a pipette with an opening small enough to put inside a cell. And ions can pass through that hollow, and you can compare the concentration of ions outside the cell and inside the cell. And as it passes through, we can read the voltage similar to that little video that I was playing earlier. So we need to have the different numbers of ions inside the cell and outside the cell. Okay? First of all, we have a different number of ions. That's a concentration difference. And that affects diffusion, right? The higher concentration on one side, ions will diffuse to the other side if they have a channel to pass through, just by numbers. So if I have this open, and I have more positive ions out here, more sodium ions out here, and fewer sodium ions out here, the direction of the concentration diffusion is going to move it into the cell. I have more sodium. Now let's say, however, I have a ton, well, not literally, but I have a lot of potassium ions. Sodium's still going to want to move down its diffusion gradient, right? However, we now bring in an electrical problem because positive is repulsed by positive. It's attracted to negative. So there's a limit. If I have a lot of potassium positive ions on here, I'm not going to be able to bring in enough sodium to reach equilibrium with my sodium if I have too many potassium already here. Because the positive potassium will repel the positive sodium. So I may not reach equilibrium with my sodium versus sodium because of the already positive ions that are there. Does that make sense? So that's the electrical, positive repelling positive. If I have a lot of negative charges in here, then I may actually be able to bring even more sodium because they're attracted negatively to the negative charge. They're attracted electrically to the negative charges. Okay? So kind of as a background. So now let's look at three factors that provide for the creation and demonstration of a resting membrane potential. This has not changed in a given cell. And so when we measure it, we create a graph that looks like this. This is 0. That would mean there was no difference in charge. I would have the same number of positive and negative charges on either side of my cell. In a nerve, in most of our typical nerves, the resting membrane potential is a -7. The minus means I'm comparing the inside of the cell to the outside of the cell. So right along the plasma membrane, as you can see here, I have more negative charges or fewer positive charges than I have right outside the cell membrane. So you can see all the positives here and all the negatives here. Okay, so we're just talking about right at the cell membrane. We're not talking about 2 millimeters away. Right at the cell membrane, I have more negative or fewer positive charges on the inside of the cell and more positive charges on the outside. So that's why I have the negative. If I had a positive, it would mean the inside of the cell is more positive than the outside of the cell. The negative refers to the number of ions that would cross to bring this to 0. Because remember, a flow of charged particles creates an electrical current. So if I were to add additional channels or open all the channels and allow everything to get to equilibrium so that my resting membrane potential is 0, it would take enough charges flowing into the cell that would create a current of 70 millivolts. Okay? Because remember, a flow of charged particles is electrical current. So enough positive charges would have to flow into the cell that I would be able to measure that electrical charge, that electrical current at 70 millivolts. All right, now what creates that? Primarily 3 situations. First, is I have large negatively charged proteins. You can see those diagrammed in your lecture notes in that previous picture. So I have a large proteins. These are too large to leave the cell membrane, to cross the cell membrane without exocytosis. And they have a net, they have positive and negative charges, but they have a net number of negative charges. That's going to make the inside of the cell more negative than the outside. Proteins inside the cell have a net negative charge. That's something that helps to make the inside of my cell more negative. All right? Not all the way to minus 70, but part of the way. All right, now I have leak channels. We talked about those earlier. And if I put in a potassium leak channel, as you saw in the video, what's going to happen to the inside of my cell? I'm going to add positive ions. That's not going to make the inside of my resting membrane potential more negative. But what does? I have more leak channels for potassium than I do for sodium. And so my net effect of this is a loss of positive ions. So due to the higher potassium permeability, I lose more potassium ions, I lose more positive ions than I gain. There's an increase, a greater number of potassium leak channels than sodium leak channels. That's just another way of saying potassium has a higher permeability. Therefore, I have a net loss of positive ions. Yes, I am gaining some positive because sodium is coming down its concentration gradient, but I'm losing more potassium than I'm gaining. Okay. It's like having pulling the plug on the bathtub and turning on the faucet, but the faucet's just coming as a dribble, whereas the plug that I pulled is a great big hole. I'm going to lose more water in my bathtub than I gain. Using that same illustration, the same thing is going to happen as a result of sodium-potassium pumps. Sodium-potassium pumps move sodium and potassium at the same time, but remember they are antiports. They're going to move potassium back into the cell and sodium out. Why doesn't that negate this effect? If I'm moving potassium back in and sodium out, why doesn't that counter this right here? It's still a net loss of positive ions. You guys remember the ratio? In my sodium-potassium pump, I lose 3 for every 2 potassium that I gain. So there's a net loss of 1. So I continue to lose more positive ions than I gain. And these 3 components, the negative charges on the protein, a faster rate of potassium loss than sodium gain, and a slightly higher rate of sodium loss than potassium gain with my pump. All of that helps to maintain negative charge. Now this isn't showing completely minus 70 because it's not showing the proteins in there. So the red channel is the sodium-potassium pump. Potassium's leaking out, sodium's leaking in, but the net effect is the inside of the cell stays more negative. Okay, now if I had the protein in there as well, then this would be almost all the way over to minus 70 with their additional negative charges. Okay? So that creates a resting membrane. When the cell's not, the neuron's not being stimulated, I haven't opened any voltage-gated channels, right? I haven't attached any ligands, I'm not opening those. When nothing else is happening and the cell is at rest, my resting membrane potential is at a minus 70, right? Now let's see what happens when we open a voltage-gated channel. So these are for, this is potassium and sodium. Notice what's happening to my voltage meter and that graph up there in the corner. So it was at a straight line. Let me reset it and do that again. I'm adding potassium ions. So my resting membrane potential, what's happening to the inside of the cell? It's becoming more positive. All right? Now, what's happening is shown here. Here's my resting membrane potential. I open the gated channel, allow positive ions in. I'm becoming more positive. So I'm getting closer to the number of positive ions outside the cell. This is known as depolarization. Getting more positive ions. I'm polarized because I'm at minus 70. As I get less, maybe minus 60, maybe minus 50, I'm getting closer to 0. So I'm becoming less polarized. Voltage gated. Well, no, this is ligand gated. All right, now I'm going to open the potassium gated. I'm losing positive ions. So the inside of the cell becomes more negative. All right? If I do that while I'm already have positive ions inside the cell, and then I lose positive ions, this is known as repolarization. Or as in the chart, I go beyond minus 70. That's hyperpolarization. I'm even more polarized than I was with the resting membrane potential. Okay, let's stop there. Tomorrow we'll talk about how this is a graded potential. We can become just slightly, so if we only open a few channels, we may just do that, or we may open a lot. We can have different degrees of depolarization. So we'll come back and start at this point tomorrow. Nobody wants to leave? No, it's usually you're out the door and it's just like, where am I supposed to go now?
My brain is completely fried right now.
Okay, so depolarization, gaining depolarization is a solid line.
Well, depolarization is the inside the cell becoming more positive.
And then we did repolarization. Inside the cell becoming
Inside the cell.
And the repolarization.
Repolarization is becoming more negative, back to where we were originally. And then if we become even more negative than that, then our cells are whole.
Inside the
Then
cell
we're in the inside the cell as well. This is All of this is referencing inside the cell.
Repolarization is becoming negative.
More back to the original, back to resting.
Negative cell.
Right. Yes.
I hear you. I'm sorry, Megan. Hold on. And hyper is more negative than negative cell.
Yes.